Hi everyone. Welcome to our bioCADDIE webinar. And in today's presentation, we're going to have Dr. Ben Busby. He is the Genomics Outreach Coordinator for the National Center for Biotechnology Information and the Chair of the Bioinformatics Department at the Foundation for the Advanced Education in Sciences. His main interest is in the integration of large genomic data sets, specifically in the metadata harmonization and statistical normalization. So please welcome Dr. Busby.
Great. So thanks, Cleo. And I'm pretty excited to be here and talk to you guys. All right. So the thing I really want to tell you about today is NCBI BioProject. So I assume that many of you probably use NCBI, at least our PubMed resource. And but not a lot of people know about BioProject. BioProject is a unifying resource that really summarizes a lot of our databases, and it summarizes them by project. So that can be, if you might think about that, it's a paper, a specific aim of a grant, or a whole grant. And I'll talk about grant integration in just a little bit. One of the reasons I'm so interested in presenting BioProject to this audience is because I really think BioProject will integrate very, very well with many of the data discovery initiatives that are happening right now. In fact, I spoke to Jeff Grethe, I guess a couple of weeks ago, and it was really interesting to see that he had integrated GEO and to some extent NCBI taxonomy. For those who don't know what GEO is, that's the Gene Expression Omnibus. It's our database of microarray and RNA-seq data. They had integrated that with his platform, but we suggested to go further and integrate BioProject. And the other reason that I'm really interested in the integration of BioProject in data discovery initiatives is we have an aspect, or we have a platform called LinkOut where we can complete a data cycle. So whether people come in through BioProject or through bioCADDIE, they can get back to the other one. And I'll describe that a little bit towards the end. But before we launch in, and by the way, this should be a reasonably short presentation. I'm thinking about 30 minutes, so we should have plenty of time for questions. I want to talk a little bit about my view of principles for data transfer. As many of you know, metadata is not particularly big, although there are obviously many problems with matching and clustering and so on and so forth, which many of you guys work on. But genomic data can be very, very large. In the SRA, which is our sequence read archive, which houses next-generation sequencing data, we have about 4 petabases of publicly available data, and we have about another 4 petabases in dbGaP, which is our genotype-phenotype database. So one thing we'd like to avoid is people copying large parts of those databases. And so we've really set up our data structures in a way to prevent people from doing that. And I think really BioProject is very advantageous in that it facilitates people coming in from other places or searching BioProject to find the exact data sets they want, and then they can actually just grab small pieces of that. We do a lot of faceting on BioProject. I'll show you an example on the next slide. Right now we use something called entree faceting, but we're moving to solar faceting for many of our resources. I'll show you an example of that later in the talk. When we integrate other data types, and you'll see that many data types are integrated in BioProject, we like to use non-ambiguous identifiers. Also with LinkOut, we have non-ambiguous accession numbers, and that's one thing that I think is really important with data integration is that everybody uses the same non-ambiguous identifiers, and I'll talk about that in a little bit. And then one way we've really designed a lot of our databases is to really encourage users to transfer not whole data sets but small amounts of data. And we've made our SRA a vertically integrated database that allows slicing of genomes and allows people to take just the parts of the genome they're interested in or they can go in through the kernel and stream the data to themselves without actually caching it on their system. One thing I'm very interested in is the democratization of data. Most of the people I work with that are interested in genomic data, many of them have never seen a blinking cursor or command line interface, but 97% of them have done something called a blast search. And so one thing we've been able to do, and I'll just use this as an example, is make our genomic sequences queryable by blast searching. Blast, for those of you who may not know, is a very, very popular, actually the most popular sequence search algorithm. And then I think it's really important for investigators to be able to report their data and to share their data. And so that's, I think, an interesting thing. And then also to link between their sequence data and other types of data, which is what you guys are interested in. And like I said, I'll talk about the LinkOut mechanism at the end and how I think the LinkOut mechanism is being used and could even be expanded to assist the bioCADDIE initiative. So with that, I'd like to just dive in and show you guys BioProject. So once again, if I want to search NCBI for something like mouse, if I search all of NCBI, I'm going to get millions of hits to all kinds of things. But if I search BioProject, I can get these things summarized by study. By the way, BioProject as a whole is growing very rapidly. In the last 4 months, we've seen 11% growth in mouse hits for BioProject, for example. But also, all of these studies are faceted by many things: project types, data types, etc., etc. I know these data types are pretty small, but you can see, for example, you can look at metagenomes, metagenome analysis, and several other types of data. I'm going to talk about a couple specific ones today. I mentioned that SRA is where we keep raw sequence reads at NCBI. So if we were to select SRA and transcriptome, we would essentially get RNA-seq data. RNA-seq data is very hot right now. A lot of biologists are really interested in getting RNA-seq data for a variety of reasons that are a bit beyond the scope of this discussion. But as more evidence of that, in the last 4 months, the amount of mouse RNA-seq data in BioProject has increased 41% in terms of the number of studies registered with BioProject. So that's obviously quite fast in terms of an NCBI database and growth. So once again, if we select SRA and transcriptome, we can immediately get all RNA-seq data that are all projects related to mouse that have RNA-seq in them. Similarly, we could do the same thing for epigenomics. So we can look at that. And then if we wanted to go further, we can look at one of these specific data sets. And the interesting thing we can see is we can get the raw genomic data in SRA. We can link to the PubMed abstract that's linked here as well as at the bottom. And also we can get the analyzed data in terms of geo. So this is a really nice umbrella way to look at these things. Also, as I'll show you later, there's a link-out mechanism so we can link out to other types of data sets and other repository-type projects. The other really nice thing, and when I talk to genomics professionals, they really appreciate this facet of BioProject. BioProject has its own abstract, but often this abstract contains key terms about the technical metadata of the project. Now, those of you interested in natural language processing type stuff, this may be a very, very interesting way to be able to cross things with metadata. I've had some very interesting discussions with folks at NIST about looking scraping metadata out of some of these abstracts and possibly comparing across that axis. So if that's something you're interested in, I encourage you to contact me. Also, in general, if people have questions about this type of stuff, please feel free to contact me at ben.busby@nih.gov. I'm also pretty Googleable. I tend to be pretty easy to find. Anyway, so we have this technical abstract for BioProject. There tends to be a wealth of information about the exact type of experiments and hopefully the preparations used for the biological samples in those experiments. So remember, this is also linked to the general PubMed abstract in many cases, which describes the actual study that was done with this particular data. So as you can see, there's a pretty big difference between the BioProject abstract and the PubMed abstract. Going back to BioProject, we can click on some of these specific data types. And what I'd like to show you very quickly is the SRA data type. So in SRA, once again, this is the Sequence Read Archive. And for genomic data that is aligned to a particular reference genome, we're able to keep this in a compressed, vertically integrated store. Now, what we're doing with SRA is we're making it available to third-party apps so that people don't have to use the command line to directly slice this with the SRA toolkit. And some of the third-party apps that, or are apps, that are available to use through the SRA toolkit are HiSat, which is a high-speed RNA-seq mapper, BLAST, which I'll show you in a minute, and we've just enabled the popular variant caller GATK to work directly with SRA data. And we're going to show that to lots of people at ASHG coming up in a couple of weeks. So if you're interested in that, please come check us out at ASHG. Right now, I'm going to show you how to search for SRA data coming from the BioProject. So the nice thing is, once you have all of these things from a specific BioProject, remember this is a specific epigenomics run we've been looking at, users can then go in and they can solar facet it based on a whole bunch of metadata fields. And all of this metadata is available. It's all scrapable. You can get it from our FTP site. There's also third-party apps that scrape and redistribute this, such as Bioconductor's SRADB. So once again, you can scrape this. Any sort of data linking database could latch, as I alluded to earlier, could latch on to sort of any aspect of this metadata and jump into that. So that's a possible thing. And as you can see, we've solar faceted it here. So that's how we use metadata. The nice thing about this type of solar faceting is you can get an accession list and use that to loop over small parts of large data sets instead of downloading all of the data. I know that's pretty complex. That said, one thing we've done to really enable regular biologists or biologists who are not computational biologists to use this kind of data is we've made this amenable to BLAST. So BLAST, like I said, somewhere on the order of 97% of biologists who are interested in genomics have used BLAST at some point in their lives. And so one can select several SRA records, send them to BLAST, and do a regular BLAST search just like you would on other NCBI functions. And that's something I'm particularly proud of because I believe this really brings genomics to everybody. In addition, several use cases for this, one could use this kind of BLAST to scan for viral genomes, either in a metagenomic sample or in human genomes, say you were interested in endogenous retroviruses. You could also use this to scan for something like a metagenomic time course. So rather than downloading all of the data and mapping it, you could search through the genomic data very, very quickly, especially once you had narrowed down the sets you're interested in by parsing the metadata. And we get a regular BLAST output. The other thing we've been able to do, we've released this in beta. You can look on Twitter. There's a lot of Twitter talk about this. You can get this data as regular BLAST output, and now we are also enabling SAM output, first on the command line, and then we'll probably do that in the GUI realm as well. If you have questions about that, please contact me offline. I'd love to talk to you about it. All right, that said, going back to responsiveness, I think in my personal opinion, we now have the genomic data sharing regulations. And so what we've done really to help out program officers at NIH is we are enabling BioProject to be tied to grant numbers. So this could be used for reporting for genomic data sharing requirements. However, where I personally, and this is my personal opinion, where I personally envision this going in the future is I think we may start incentivizing people to produce high-quality data sets by allowing people to put that on their grant renewal applications. And so I think through a mechanism like this, one could see how many people accessed what data sets. And that's to me something very, very exciting. If we can monetarily incentivize people to produce good data sets. Once again, my personal opinion. But this is something that we are currently enabling. That said, this comes from the bioCADDIE white paper. I think we want to help in terms of enabling this user-friendly means to find relevant data. And I really think that BioProject could be part of the puzzle from the sequence data side. And I'd really encourage the bioCADDIE project to link more to BioProject. And also, I'd like to introduce you guys to this concept of LinkOut, where we could really integrate this into a larger data discovery framework. So some of you, if you're involved with medical libraries, may be familiar with the concept of LinkOut. And what LinkOut, one of the things it does in the publication sense is it allows specific libraries with specific subscriptions to offer full-text links to their users on the PubMed page. But we also have LinkOut for non-bibliographic things. And I'll tell you a little bit later, we're going to offer a webinar on this specifically on October the 2nd. One of the things we can link out from is BioProject. So if there were a large project like bioCADDIE who wanted to link to other things from BioProject to other data types, we could certainly do that. And I'd like to show you a couple of examples. One example of that is GOLD. I don't know if any of you have worked with microbial genomics, but in my personal opinion, GOLD for many years was a world leader in the collection of metadata around microbial genomes, and GOLD links to BioProject, which is very convenient. Also, what we see here is that different strain sequencing projects from the same bacterial genome are linked to one another. So I think that's a particularly useful thing. And we can see the link out. I need to show you with the actual cursor. We can see the link outs on the right. So that links to both the GOLD database as well as LSU's Silva database. Here's another example, and here the nice thing is we can see that we have an annotated genome with a whole bunch of protein sequences as well as a PubMed record. The other thing this does for a data discovery index is you have a non-ambiguous publication link with a data set, which is really nice because, I mean, I have a lot of friends who do natural language processing type stuff, and let's face it, there is ambiguity in data that comes out of natural language processing. Absolutely. I mean, that's part of the nature of the beast. And so what I would say is with this kind of mechanism, because it is submitter driven, you have non-ambiguous links. If you're interested in LinkOut from NCBI, please check out our webinar on October 2nd. Should be a 20 to 25 minute webinar similar to this one. And also, you can check out our archived webinar. There's a lot of stuff on BioProject, SRA, many of the things I talked to, including a webinar fairly similar to this one on some aspects of BioProject, but much abbreviated. All right. With that, I think I've kept it reasonably short, and I'd like to open the floor for any questions or comments that you guys may have.
The conference has been unmuted.
Yeah, I think Cleo's unmuted everybody.
Great. Thanks. This is Lucilla from bioCADDIE. It's an excellent presentation, very exciting work that is happening in there. So I would like to ask the audience for any questions, either through the chat box or through the microphone. We have a very well-behaved audience with no background noise right now. So while people raise their hands and warm up, my question may be a little bit outside of the scope, both of bioCADDIE and of BioProject, but it is related to the indexing of the biosamples, because I do have, for example, many postdocs or other researchers looking for where can I find multimodality data on the very same sample. Is that something that through your biosample indexing you would be able to find, or is there any other way?
Well, so interestingly, after I talked with Jeff, which I mentioned, I went and talked with Tanya Barrett about this exact thing. And it turns out that Tanya Barrett is actually on the line right now. So actually, I think I'd like to turn the floor over to her and hear her thoughts on that, if she doesn't mind me putting her on the spot.
Hi, Ben. Hi, everyone. Right, so actually at this time we do have the capability to allow submitters to point different types of data to the same biosample if that biosample was reused in their studies. The challenge is getting submitters to actually do that, so there's probably lots of examples that are not pointing to the same biosample where they could be, but the capability is there. We also in Biosample have an ongoing project to cluster, for example, well-used cell lines, like commercial cell lines that lots of different people use. There is plans to create master biosample records for those, to which all the data generated from that sample can point to. So these are work in progress areas right now.
Excellent. Thank you very much.
Sure.
So Ben, we have one question in the chat box. I think you can read that from Mary Mangan.
I absolutely can, and I've been sitting here thinking about the answer to that question. We could potentially link papers, and we do have the related publication link. So there's 2 steps right now to that. So if you go to the actual publication, you could click on related publication. Also, from many of our resources, you can link to related publications. I don't think there is a BioProject citation link, but that may be in the works right now. Tanya or Kathy, did you have any sort of more information on that?
I'm not aware that BioProject is going to do that, but I think it might be possible. It's certainly technically possible for us to implement a similar BioProjects based on the PubMed similar studies. So we already do that in Geo, where you can find similar studies based on the linked PubMed links. So I will bring that up with the BioProject development team.
Right. I just wanted to go back for a second. And by the way, thank you guys for the suggestions. I wanted to go back and read the original question, which from Mary Mangan, because I have been informed by a couple of you guys that you guys can't see them. And that is, and Mary says, could you possibly link papers that discuss a project back to the project? I mean, people who use the data downstream, not just submitters. And so that's what I was saying. And Tanya reiterated that there's a related papers link, and in theory, we can link that directly from BioProject. And we'll talk to them about that being a potential use case. And speaking of potential use cases, if you ever have use cases about molecular databases at NCBI or aspirational use cases, please feel free to email me, because those are the sorts of things I collect and discuss with our team leads.
Great. Yeah, we did have, or still have in bioCADDIE, an effort just to collect these use cases, which we definitely can share with you. Some are more immediately resolvable. Some are for the future, of course, but I think there are lots of interesting sides of it. Because my other interest is in human subjects, so that's related to the biosample, because many of the volunteers tend to volunteer for multiple studies and be measured on different things, of course, at different times. But that would be also an interesting feature if we could have, by the same mechanism, link biosamples from the same individuals and over time. But again, that's probably a use case for the future. Maybe that would come with the precision medicine initiative, so if we might not be that much of a future far away that we would think into. I don't see any other questions from the audience. Please, folks.
I just wanted to make one small note on what you just said. So dbGaP is also integrated somewhat into BioProject. So if you were doing a fairly specific BioProject query, not so much through BioSample, but with BioProject, you could use that to integrate some of the studies in dbGaP if you had a very specific phenotype.
Right, but could you link two records from different studies without going through the data access committees and so on, even the process of linking them? And I don't think, if I remember well, that they do have a sample ID, do they?
For particular individuals?
Yeah.
As far as I know, no, we do not have that. But yes, you're right. I mean, people do volunteer for studies, and that's something I will certainly bring up with Mike Fiolo, who's the lead at dbGaP for NCBI. Also, a lot of what happens at dbGaP goes through the NIH Office of Science Policy. So they do sort of all the policy stuff, they run the DACs, all that sort of stuff. We do the technical side of dbGaP.
Yeah, because I've heard, that there could be discussions of unique identifiers for individuals, which again is a very long discussion probably, but for volunteers, I think that could be something that would be of immediate value.
Yeah, I mean, I know that's something that several groups are working on, coming up with sort of global identifiers for patients. I know the autism community has been doing some work in that space as of late. And certainly it's something we've talked about.
Okay, any other questions for Dr. Busby? Okay, so if we don't have any other questions, I would like to thank you, Dr. Busby, for this great presentation today. And just to announce, our next webinar is going to be on October 8th in a different time because our presenter is from Australia, so we have to adjust the time zone. So it's going to be at 2 p.m. Pacific time, and Dr. Ariani is going to present the research data switchboard. Okay, thank you, everyone.
Thank you all.